Okay, now let's pick up and start talking about the immune system. So the immune system itself is meant to protect our body from pathogens and things in the environment that we are exposed to. So pathogenic or pathological bacteria, of course. So that would be a bacterial infection. Foreign tissue cells and cancerous cells. Our body, over the course of our lifetime, does develop cancerous cells, but our immune system actually gets rid of them. So that's a great thing, right? So what is the immune system? The immune system itself is made up of specialized cells and molecules that protect us from things that are bad for us, things that are pathogenic or that'll make us sick. So there are two types of general immunity. There's nonspecific immunity. These are just barriers, okay? Skin, our integumentary system, is the most obvious mechanical barrier to bacteria and other harmful things in the environment. But we also have like tears and mucus. These are fluids that wash bacteria away, and believe it or not, our tears have an enzyme that can kill bacteria. So our tears and mucus are very effective at trapping and killing bacteria. We also have inflammation, which brings immune cells, which we'll talk about later, B cells and T cells, and macrophages, to an injury site. So what inflammation is doing is, let's say, for example, you get a cut. There's an inflammatory series of chemical messengers that are triggered to increase after the clot forms, to increase localized blood flow to the area, to increase vascular permeability. What does that mean? So remember back to the last chapter where we talked about endothelial cells being basically the capillary, what make up capillaries. It's like endothelial cells with some basement membrane around it. Well, when we have inflammation, our vascularization, the area, becomes swollen, and as it stretches, it makes those capillaries more permeable. There are other signals that cause an increase in permeability of the capillary beds. Don't worry about that right now. But what that does is it promotes the movement of white blood cells from the bloodstream out into tissues. So let's say here is a capillary, okay, and we have these endothelial cells that are kind of sitting next to each other, and there are little gaps. White blood cells, which I'm going to identify as blue here, white blood cells sneak out into tissues. Okay, so this is going to be, I'm just going to make a circle for a white blood cell. They sneak out into the tissues through a process called extravasation, and now they can go and eat up. Let's say you have, let's make that a different color. We don't like it blue because that's our white blood cell. Let's say now we have a bacterial cell. The white blood cell, a macrophage in this case, can come over and engulf it and phagocytize it. All right. So this whole process of inflammation is a good thing. It increases blood flow, allows extravasation to occur, allows these white blood cells to interact with tissues and cells that are going to be potentially harmful. So here's the pathway of an inflammatory response. Bacteria, and this is inflammatory responses relative to bacteria. Bacteria enter the tissue damage occurs. The tissue damage causes mediators. Mediators are going to be those regulatory proteins that are released that attract white blood cells to the area. It's called chemotaxis, just for fun. Increased blood flow, that's our inflammatory response, where you have that cut, and then once it's infected, right, it feels kind of warm. That's because you have increased blood flow to the area, and increased vascular permeability, which enables extravasation of the white blood cells into the tissues. That increases the number of leukocytes, white blood cells, and chemical mediators to the site of tissue damage. Let's go back to red here. Okay, the chemical mediators. That hopefully will destroy bacteria, and the extra white blood cells that are there will phagocytize the dead bacteria. If no bacteria remain, tissue becomes repaired, and ta-ting, we're good. If bacteria remains, well, then we have continuous inflammation in the area until hopefully the bacterial infection has been taken care of. So in terms of immunity, what's going on in the body. We just talked about simple inflammation, and we talked about non-specific immunity, which are going to be those things, those barriers, our tears, our mucus, and so forth. Moving on to specific immunity, this is where our body learns to recognize and respond to harmful compounds, harmful viruses, and harmful bacteria. This is where our immune memory is, specific immunity. Now, there are two types of specific immunity. Number one, I'll put a number one here instead of a dot, is inherited or inborn immunity. And this is where we have, as humans, immunity conferred upon us against certain diseases and stuff from birth. How does that happen? We'll talk about it shortly. So this, let's come back here. This inherited immunity can also be called acquired immunity. Now, this is natural immunity that is due to exposure to a causative agent. Active is when our own body does this. Okay, so active is self. But the passive, if we go back here, is our inherited, inborn immunity. Passive immunity happens when antibodies pass from the mother to the fetus through the placenta or through mother's milk as the child is nursing. And so inherited immunity is a big deal. Right now, we know with COVID-19 that pregnant mothers who have been exposed to COVID-19 and have survived the illness are passing their antibodies to COVID-19 onto their newborns. That's a big deal. What that means is that these babies are being born with maybe not, we don't know how long that immunity is going to last, but we do know that they have acquired immunity now through passive mechanisms. So they have inborn, inherited immunity. Now, the rest of us get to go and get vaccinated if we haven't been exposed, because obviously we're not fetuses, or we weren't recently fetuses. So the rest of us have to get vaccinations. And so this is an artificial immunity, right? So active immunity as a result of vaccination is important because now our bodies are trained against this agent that we are deliberately exposing ourselves to, right? Because we're getting the vaccination. Let's go back one more slide. Now, what about, for example, passive artificial immunity? Well, this is a situation where, unlike when we're being vaccinated against something in the environment, somehow material from another individual's immune system gets into the non-immune individual. This particular case, we'll run into in a little bit in terms of a situation where somebody were to receive, let's say, a tissue transplant. So it's a protective mechanism to ensure that our body's immune system isn't, or our body isn't being contaminated with another individual's cell type. All right, so what's going on in terms of how the immune system uses what we're going to call antibodies to protect us? So antibodies are proteins with very specific sites. So an antibody actually looks like this. It looks like a Y shape. Ooh, that's like the ugliest Y shape I've ever seen in my life. So I have to go get my eraser here for a second. Oh, the days of actually having a whiteboard. I miss those. So it's more of a Y shape like this with little strings in between. And then these disulfide bridges holding together these two long strings. So these are the heavy chains of the protein. These are the light chains. And this region over here that I'm going to highlight in blue, this is called the variable region. So in other words, these two tips are specific to combine with something else in the environment. All right, what could that possibly be? Well, in the environment, cells that aren't a part of your body, so let's say we have, I'm going to make this a different color because I want to make this an invading pathogen. So let's make our pathogen orange. Let's say, for example, we have an E. coli cell. that E. coli cell might have little proteins on the outside that look like rectangles. Now, one of the things I want to mention to you right now, to not confuse, is we have words for the proteins, we have names for the proteins on the outsides of cells, all right? In this particular case, we're going to call this protein the antigen, okay? And this whole structure here is the antibody. Antibodies sometimes are also called this. It looks like the little fish shape. That just means anti, okay? An antibody is going to hook onto an antigen. So I'll show you another picture in a few minutes. But what happens is that antibodies produced by our immune system actually hook onto the antigens on the surface of a cell, kind of like a sticky note, all right? So let's say, for example, we somehow get E. coli under our skin. Yuck, right? And the E. coli has this little square that is going to fit into this site on an antibody, all right? Let's say you have another antibody here, and I'm just going to draw it as a Y shape right now, okay? And it hooks right there onto the end, to the variable region of the heavy and the light chain, all right? So here you have a variable region hooking up to, it shouldn't look like a Y, a variable region hooking up to an antibody. What this does is it causes the E. coli cell to become tagged or labeled. It's like putting a sticky note on it. When antibodies are stuck onto an invading pathogen or something, that signals the body to neutralize whatever that is. It allows it to neutralize toxins. It can allow these to clump up together to do an agglutination reaction to bring a bunch of cells together, and best of all, it promotes phagocytosis. So what this humoral immunity does for us with antibodies is it comes together. If you get a bunch of bacterial cells under your skin, it brings them together as a clump and promotes phagocytosis. So here's a picture where you have an antibody and the antigen. So let's just pretend that little rod in the middle is our E. coli with two proteins on the outside. Oh, I got skipped forward real quick. Sorry about that. Go back again. So we have our antibody here, our little E. coli here with the antigen on the outside. The antigen and the antibody come together to form a complex that can activate the complement cascade, which is a series of steps that'll help kill the E. coli. It definitely allows an inflammation response to happen. There's an attraction of other white blood cells like macrophage here. And it allows macrophage and other B cells to start secreting histamine to promote an inflammatory response. So all of these steps together, it's actually a lot more complicated than this. If you take physiology at Merced College, we go over this in much more detail. But the whole idea is that these antibodies stick to and label specific pathogens based on the antigens on the surface of the pathogen, and it targets that pathogen for death and destruction and a phagocytic event. So we'll be moving on to the next part, the next slide, immune system molecules, in the next video. Complement system is so totally cool.